Welcome to you in new video of Amar Biotech. In the final stage of protein synthesis, the nascent polypeptide chain is folded and processed into its biologically active form. During or after its synthesis, the polypeptide progressively assumes its native conformation, with the formation of appropriate hydrogen bonds and van der Waals, ionic, and hydrophobic interactions. In this way, the linear, or one-dimensional, genetic message in the mRNA is converted into the three-dimensional structure of the protein. Some newly made proteins, both prokaryotic and eukaryotic, do not attain their final biologically active conformation until they have been altered by one or more processing reactions called post-translational terminal modifications. The first residue inserted in all polypeptides is N-formylmethionine, in bacteria, or methionine, in eukaryotes. However, the formal group, the amino-terminal methionine residue, and often additional amino-terminal, and, in some cases, carboxyl terminal residues may be removed enzymatically in formation of the final functional protein. In as many as 50% of eukaryotic proteins, the amino group of the amino-terminal residue is N-acetylated after translation. Carboxyl terminal residues are also sometimes modified. The 15 to 30 residues at the amino-terminal end of some proteins play a role in directing the protein to its ultimate destination in the cell. Such signal sequences are ultimately removed by specific
amino acids. The hydroxyl groups of certain serine, threonine, and tyrosine. Residues of some proteins are enzymatically phosphorylated by ATP monomethylene. Dimethylysine residues occur in some muscle proteins and in cytochrome C. The calmodulin of most species contains one trimethylysine residue at a specific position. In other proteins, the carboxyl groups of some glutamic acid residues undergo methylation, removing their negative side chains. The carbohydrate side chains of glycoproteins are attached covalently during or after synthesis of the polypeptide. In some glycoproteins, the carbohydrate side chain is attached enzymatically to asparagine residues N-linked oligosaccharides, in others to serine or threonine residues O-linked oligosaccharides. Many proteins that function extracellularly, as well as the lubricating proteoglycans that coat mucous membranes, contain oligosaccharide side chains. A number of eukaryotic proteins are modified by the addition of groups derived from isoprenyl groups. A thioether bond is formed between the isoprenyl group and a cysteine residue of the protein. The isoprenyl groups are derived from pyrophosphorylated intermediates of the cholesterol biosynthetic pathway, such as farnesyl pyrophosphate. Proteins modified in this way include the ROS proteins, products of the ROS oncogenes and proto-oncogenes, and G proteins, and lamins, proteins found in the nuclear matrix. The isoprenyl group helps to anchor the protein in a membrane. The transforming, carcinogenic activity of the ROS oncogene is lost when isoprenylation of the ROS protein is blocked, a finding that has stimulated interest in identifying inhibitors of this post-translational modification pathway for use in cancer
chemotherapy.
Many
proteins are initially synthesized as large, inactive precursor polypeptides that are proteolytically trimmed to form their smaller, active forms. Examples include proinsulin, some viral proteins, and proteases such as chymotrypsinogen and trypsinogen.
After folding into their native conformations, some proteins form intrachain or interchain disulfide bridges between cysteine residues. In eukaryotes, disulfide bonds are common in proteins to be exported from cells. The cross-links formed in this way help to protect the native conformation of the protein molecule from denaturation in the extracellular environment, which can differ greatly from intracellular conditions and is generally oxidizing.
15 to 30 residues at the amino terminal end